in nano delivery, a nano system that either carries existing active ingredient, but also with the potential to consider alternative active ingredients, pheromones, or genetic materials such as those that are considered here. So a lot of really exciting opportunities with a wide range of nanocarrier system. I've just put a couple of examples here that are very often discussed in literature. Nanocapsules made of polymers, dendrimers, hydrogels, fibers that are electrospun over fields, porous silica, and so on. The main objective of those nanocarrier systems, from my perspective, is to address the weaknesses of current plant protection products. And this can occur at different stages of the pesticide application. This is what is summarized in this scheme. The application stage may be the main focus when the nanotechnology helps achieving better stability of the formulation suspension or achieving a better, more homogeneous application onto the crop. Maybe looking at mainly reducing those 90% losses that may happen through transport processes or degradation processes. Or we may be looking at improving the biointeraction by increasing bioavailability, controlling the release or targeting the release to really minimize the uptake of non-target organism of these active ingredients. So overall, these examples to me sound like nanotechnology has really the potential of reducing application rate or application frequency, which is an excellent thing because it may overall decrease the environmental footprint that conventional agriculture has on the environment and indirectly on human health. But it's not the opinion of everyone, and what I have observed when screening the literature is that research is really compartmented, and by looking at literature generated by different communities, one can get a very different perception of what nanopesticides mean, what they are, and whether they are nano good or nano bad. So I've been talking to two main communities over the last year, pesticide community, nano community, under the umbrella of environmental scientists. And this is amazing on how the opinion is different from one community to the other. So the nano community basically is not really used to the idea of applying toxic compounds in the open environment, and they tend to consider nanopesticide consisting of titanium dioxide, nanosilver, carbon nanotubes, and this is shocking to me as well if someone think of applying those compounds or those particles in the open environment. Actually, the use of nanopesticide in this context will represent the only intentional diffuse input of engineered nanoparticles into the environment. And that sounds really concerning. So as a consequence, this community tend to consider nanopesticide as emerging contaminants. The pesticide community has a very different opinion, and they tend not to be so excited about the technology because they are aware that nanotechnology hasn't really supported the development of a new way of controlling pests or a new mode of action per se. They know also that regulators will never let farmers apply carbon nanotubes or nanosilver. They are aware of constraints of the market, and therefore, from my perspective, they have a more reasonable approach of the product that may reach the market soon, which consists of formulation technology. And formulation technology is known for a long time as one way of mitigate the risk associated to conventional agriculture. So some potential, but not necessarily nano bad, rather a way to improve current product. Here at the bottom of the slide, I've placed the regulators or decision makers, who are the people who decide whether a new product can enter the market or not. And they may be confused hearing those two very different messages related to the applications of nanotechnology in plant protection. Should they only focus on the risk? Should they also focus on benefits? Do we have tools to evaluate the benefits that the new products represent compared to conventional products? It appears that no tool exists. And this is where my research intervenes, and this is what I'm aiming at, producing some tools that will help evaluators as well as people developing those products to really develop the best product that will represent the more benefits and the least risk as possible, but based on scientific data, first principle, and not only perception. One question that is important at this stage to ask is how many of those products are actually reaching the market. Because if I really want to help regulators, I may want to focus my work those products that are really in the pipeline. And we may also ask ourselves whether regulators really need these products. If we are really at a very early stage of development, we may not worry and rather focus our attention on other questions. So these are 3 examples taken from different parts of the world and that indicate nanotechnology-enabled products for plant protection are definitely reaching the market. And the first, looking at North America, a company called Five Crop Protection that I've been working with over the last years, who has developed an allosperse polymer-based enabled plant protection delivery system, and they have obtained authorization to place a number of their formulation onto the market in the United States. The second example is taken from my home country in France, where I read a report where the data is collected from companies. Companies have to declare the nano products that are either produced, imported, or distributed in France. And every year, all the data is collected and some data analysis is carried out. And one of these graphs is showing the sector in which those nano product is used. And we see that plant protection product actually arrive first with about 800 products. That seems a lot to me, and the numbers probably have to be taken with a lot of care. But this indicates to me that industry is definitely working with this technology. And the last example is an Australian example. You may know, perhaps, or have heard about the company called Star Pharma. I understand they have been communicating quite a lot about some field trials carried out with dendrimers technology and glyphosate, showing really high efficiency compared to conventional product. And I understood they were really close to submitting a dossier for regulation. So in all parts of the world, I think regulators are really in a need for tools to take the right decision. So this is an overview of how ecological risk assessment works in most parts of the world. And pesticides are probably the most regulated compounds throughout the world. And before being authorized, they have to go through 2 parts. The risk assessment is made of an exposure assessment that looks at understanding where the compound goes, and it relies on a number of fate parameters, mainly sorption, degradation, leaching, exposure models that are pretty sophisticated and that support the prediction of environmental concentration in different compartments. So we try and predict concentration in soil, groundwater, surface water, sediments, and so on. The second part of the risk assessment looks at the biological effect. And pesticides are pretty particular. We're not looking only at the hazards, but also at the efficacy to the target pest. And this is something a bit particular about pesticides. And then we can compare the exposure, so the concentration occurring in the environment, to the level we know may be hazardous for some species. By comparing the two, can decide whether the risk is acceptable or not. Again, there's always a little bit of risk because there is always exposure, pesticides are applied, and there is always a little bit of effect. But we can decide whether the risk is acceptable in view of the plant protection that we are getting. So in view of this very sophisticated framework that has taken decades of research to put in place, it has been validated and so on, the first question I ask myself is whether this framework can be used with nano-enabled pesticides. That was the question we also asked ourselves during a series of workshops. And this is when I started collaborating with Rai Kukana. This is a picture taken at the first workshop that took place in New York in the UK in 2013. And it was very interesting to put together academics, researchers, people from industry and regulators, aiming at producing a set of guiding principles to achieve a harmonized ecological risk assessment of pesticides. The first question was, is this framework working? And we all agree that that's pretty robust, that's probably going to work. But we also all agree that we may need some additional metrics, additional parameters to describe the nano-particularity of the nanopesticides after it is applied in the environment. And this is how the schemes look like. We have the pesticide assessment here, classical exposure assessment, and from this point on I will only focus on exposure assessment. We propose to introduce the durability parameter, that is basically a measure of time, how long the pesticide is going to behave as a nanoparticle. If we imagine a capsule, a nanopolymer carrier, this is represented here, loaded with an active ingredient. Our assumption is that if the active ingredient is released immediately upon application, we probably do not see much influence of the nanoparticle. It's ending as being like another pesticide. So in case the durability is very small, we only need to worry about this element, classical pesticide. On the other hand, if the durability is significant, the time during which the active ingredient is associated with a nanoparticle is going to behave like a nanoparticle in the environment. And this means that different fate processes and first principles have to be taken into account. And this is what is represented here. Based on this basic scheme and the principle or the concept of durability, we organized another workshop that looked at trying to provide further guidance for regulators on which fate parameters they may need to ask people applying with new products, and really to provide a relative assessment of exposure of nanopesticide compared to non-nanopesticides. And our scheme looks or produced several matrices, and I'm showing you one matrix here where we basically try and combine properties from the active ingredient, properties from the nanocarrier, and from this try to guide the regulators on specific aspects they should be looking at in particular. Not asking everything every time, but to really, at the stage of problem formulation, target the specific questions that we believe are important for a given product. So this matrix works in the following way. We have the mobility of the active ingredient and the persistence of the active ingredient. These are the two main fate parameters driving the fate of chemicals in the environment. And here we have an additional dimension with the durability criteria, which is specific to nanopesticides. In some cases, we think that there is no difference between nano and non-nano pesticides. And these are the situations that are indicated by the OK flag. In some situations, we believe that persistence is going to be the main change in behavior. And this is the flag with P. And in some other situation, we believe that mobility is going to be the main factor influenced by the nano character. And these are indicated with the flags M, either enhanced transport or retarded transport. So I'm going to give you an example now on how this matrix is working. If we take again our polymer nanocarrier loaded with an active ingredient, first assuming that the active ingredient is immediately released upon application in the field. We are on this first panel where durability is 0, and we see that we only have OK flag. And we believe that the classical pesticide assessment scheme is suitable. But when we start increasing durability, we see that in almost all situations, we can expect some significant change in environmental distribution. Now, we have a mobile nanocarrier. If the active ingredient is non-mobile and non-persistent, we are locating here, the association of a non-mobile, non-persistent active ingredient with a mobile nanocarrier is much likely going to change both the persistence and the mobility. But if the active ingredient is already mobile and non-persistent, who are moving along this line, then mobility is probably not going to be the big change because the active ingredient was already mobile in the environment. And we advise regulators to really focus on persistence. So we are hoping that this scheme, which I hope I described in a clear way, can really help regulators in some situation not asking any additional data. In other situations, ask really in a targeted way some data, for instance, only related to persistency. So now I'm going to talk to you a little bit about the experiments that we have carried out in our laboratory in parallel of this more desk-based work. But I think the two schemes really work together, because the first thing that I did was to try and source series of nanopesticides from academic partners, from industry, and to test the classical fate parameters, protocols that are developed and really described by the OECD guidelines. I've been working with fairly large portfolio of nanopesticides up to now. This is just a quick overview showing that we've been considering different nano properties, so different size, different colloidal stability, either electrostatic, steric, or electrosteric, which is something that is going to dramatically affect what happens to a particle once it is released in the environment. And here are the active ingredients we have considered. The main idea was to cover a wide range of sorption and degradation. So we can really probe with some model nanopesticide the scheme that we have developed earlier with regulators and industry. So we basically determine fate properties for all those products, and this is a very long story that I'm going to make very short. Nanopesticides do not behave like conventional products. Not in all cases. Sometimes there's no difference, but sometimes there are important differences that we cannot really explain because those protocols have been developed to describe the fate of solute, not the fate of particles. So we see there is a difference, but we don't really know what the difference is due to and how it would change from one environment to the other. So we get a hint that probably we need this other part of the assessment. Instead of showing you some detailed results regarding the transport and degradation of nanopesticides generated in our laboratory, I thought that today I would show you the result of meta-analysis we recently published, and that is the first systematic comparison of the characteristics of nano versus conventional pesticides. So we collected all the data available in literature and generated box plots for different parameters looking at physicochemical properties, fate properties, as well as efficacy. And I'm showing you here some examples related to sorption. So the box plot represents all the data we found in literature, the 25th, 75th percentile, the line is the median, and the cross is the average. And these are the comparisons of nano versus conventional for sorption coefficient. So is sorption significantly different for nano or non-nano? If we look at median and average, we may be tempted to say, yes, the ratio is 1, and this is the situation where there's no difference. But we see that the box is also covering data from 0.5 up to 2, which means that transport may be affected, increased or decreased by a factor of 2, which is a great impact on the environmental redistribution of pesticide active ingredient after they are applied. This is the same box plot generated for data related to degradation or persistence in the environment. You see that the median average are very close to 1 as well, suggesting there is not much difference. But if we have a closer look at which type of degradation process we are looking at, in degradation in soil, there's probably not so much of a difference, but for photodegradation, we see a great potential of nanotechnology to decrease the photolysis of active ingredient. So overall, this data suggests to me that some great differences are possible, but it depends on the process, and it depends on the nanopesticides, which tells me that we have to apply a case-by-case assessment. So, when there is a large difference in fate, we need to integrate the durability parameter. And naively, perhaps, when I started working on the topic, I thought that measuring durability would not be very difficult. I thought I would simply apply some protocols that have been applied in the past to measure release of pharmaceutical compounds, for instance, applying centrifugation, also ultracentrifugation, filtration, dialysis. What we want is just to separate that portion of active ingredient that is released from the nanocarrier from that portion that is still associated with the nanocarrier. And we want to do this over time. That sounds pretty simple, but it appears that pesticide active ingredients tend to interact with membranes. So filtration, dialysis are not methods we can apply to a broad range of products. And it appears that many nanocarriers, especially those made of polymers, are not very dense, and it's possible to separate them by centrifugation, but it may take a very long time and very high speed, which does not help us if we want to measure kinetics at short term. An additional thought, because I'm a soil scientist by training and I'm particularly interested in what happened to nanopesticides in soil. I'm also wondering how representative those data generated in highly diluted conditions, how those data can translate to what happens in soil. Is the release of the active ingredient going to be different or not? This is something I would really like to find out. Because experimental, direct experiments were not really successful, I thought that we could apply an indirect evaluation of the durability or the release rate under conditions that were more representative of the condition that the nanopesticides is going to see after its application. So we have applied this for degradation in soil and photodegradation, but the principle is really the same. Here I'm showing you degradation curves. So we have the concentration of an active ingredient decreasing over time through a degradation process. And the data shows that the pure active ingredient here is red, degrades faster than the nano-formulated active ingredient. I think we can fairly assume that this delay in degradation is due to the association of the active ingredient with the nanocarrier, and basically the nanocarrier protects the active ingredient from degradation. In other words, release and degradation are two sequential processes. This is what is represented here. First, the active ingredient has to be released, then it can be degraded. Assuming that those two processes can be described by first-order kinetics, which is fair enough based on literature, we can use those two curves, fit them with first-order kinetics, and by inverse modeling, figure out what was the release rate and get some data that we can then further use. We have, with this indirect way, a tool or an approach to measure release under more realistic conditions. So this is where we are now. I think we have a new scheme, methodology, which I hope are useful for developers and evaluators. We are now really concentrating our work on this element that is missing, the characterization of the nano property, which is first a challenge, but more interestingly, what happens to the nanocarrier after it is released into the environment. All our work first focused on the active ingredient, which is much likely the most toxic element of the nanoformulation, but we should not forget about the nanocarrier. We may want to know what happens, especially as pesticides are applied repetitively in the same place. We need to know what happened. And this is also a very challenging question that I'm sure will keep us busy for a little longer. Before concluding, I thought I would have last slides and touch upon a last topic that looks at what do evaluators do with the pesticide fail parameters? How do we derive predicted environmental concentration from those parameters? I want to briefly mention an exposure modeling exercise I carried out 2 years ago by considering one of the exposure model that is used in the European context by regulators to measure or to predict the concentration of pesticide leaching down to groundwater, down to 1 meter. This is the regulatory threshold. And if one predicts that the concentration of pesticides may leach down to 1 meter at concentration exceeding 0.1 microgram per liter, then this is too much. This is not going to be authorized. And I use this model for 9 soil climate scenarios that are representing collectively realistic worst-case scenarios for Europe agronomic conditions, and I used 2 main approaches. The first one that I call direct inputs, basically just I took the parameters determined in the laboratory and used them as input into the model. I completely ignored the fact that the active ingredient may be released over time. And in the second approach, I integrated durability and slow release and used the model. There are some ways that we can make the model to simulate this. And again, a fairly long story, very short. This is the outcome that we get. By using directly the input without thinking of trying to represent more closely what really happens in the environment, the model predicts that concentration leaching to groundwater are increased when the compound is nanoformulated. But if we take into account the release, then we get a great decrease in the concentration potentially reaching to the groundwater. In other words, one approach gives nano bad, the other one gives a nano good. And we'll keep on working on those issues because I think it illustrates very well on how we can get different answers if we just do not ask ourselves the right question. And I think it really highlights how important it is to not only focus on the risk that nanotechnology can bring to the plant protection product sector, but also on the potential benefits. And I think that would be a pretty good benefit if we could reduce contamination of groundwater. So here's my last slide, just a wrap up of some key messages I wanted to convey today. Nanopesticides term has been used to describe a really wide variety of products that are very different stage of development, and it's important to well define what we are talking about. I cannot tell you today nanopesticides are nano good or nano bad. We need to go a little bit more into the details. I think the products that are the closest to markets are formulations and formulation technologies, something that we should probably focus a little bit more and give a bit more attention. And I think that there is a real potential for nanotechnology to increase the efficacy of the active ingredient, reduce their losses overall, just to reduce the amount of pesticide we are regularly applying to the environment. And I think this is a great potential. My research so far has focused at understanding what happened to the nanopesticide, the nanocarrier, trying to evaluate the risk and benefits from an ecological perspective, and overall looking at the environmental footprints that nano-enabled products may represent compared to other products. But this is only a little part of the puzzle, and there are other very important socio-economical factors that are really crucial to take into account. And I'm hoping that in the future, research will be more collaborative, multidisciplinary, so that the whole picture can be really considered. And here are three aspects that I think are really important to consider: the scalability of the synthesis and the production. This is something that needs to be considered. Economic viability compared to conventional product. Agriculture is a really low profit industry. We're not talking about pharmaceutical industry, and this is a big constraint into the sector. So this has to be kept in mind. And also aspects related to social acceptance. I think we can learn a lot of what has happened in the past with genetically modified organisms. It hasn't necessarily been really successful, and I hope we can get a better approach in informing the public about potential benefits and risk, and talk about this aspect in a more reasonable way. So here's my email address in case you would like more information, and I thank you very much for your attention. I'll be happy to answer any questions you may have.